Hello and welcome dear viewers to the another very interesting topic on bioequivalence. The topic is bioequivalence problems and solutions for pharmaceuticals. So in this video we will understand the basics of bioequivalence criteria, when the bioequivalence fails and what are the solutions mainly for the pharmaceutical formulations. So bioequivalence is needed for the formulations to provide the assurance that the safety and efficacy of the formulation is achieved. Generally, for the innovator formulations, bioequivalence is done with comparing the formulation before clinical studies and on the manufacturing site formulation. So that is also a bioequivalence. When there is a site transfer, then bioequivalence is required. So bioequivalence provides assurance of safety and efficacy of the formulation by meeting the pharmacokinetics of the formulation being tested against the reference formulation. So for generic formulation there is always a reference formulation, and for innovator formulation there is a formulation which is tested in clinical studies and the formulations which are manufactured at the manufacturing site on the large scale. So as per the WHO, two pharmaceutical products are bioequivalent if they are pharmaceutical equivalent or pharmaceutical alternatives, and their bioavailabilities in terms of rate, Cmax and Tmax, and the extent of absorption, that is area under the curve, that is known as AUC, after the administration of the same molar dose under the same conditions are similar to such a degree that their effects can be expected to be essentially the same. The very short definition of the bioequivalence is the two formulation can be said as bioequivalent if they are pharmaceutical equivalent and if their bioavailability, that is rate and extent of the absorption, are similar. And once the rate and absorption gets similar, then the pharmacodynamic response can also be similar, and that time the formulation or generic formulation or the test formulation becomes therapeutically equivalent to the reference formulation. Now we will see the definitions given by other regulatory authorities like FDA. The absence of a significant difference in the rate and extent to which the active ingredient or active moiety in the pharmaceutical equivalents or pharmaceutical alternatives become available at the site of drug action when administered at a same molar dose under similar condition in an appropriately designed study. So as per FDA, there should not be any presence of the significant difference, that is the absence of significant difference in the rate and extent of the absorption. Then as per EMEA, two medicinal products containing the same active substance are considered bioequivalent if they are pharmaceutical equivalent or pharmaceutical alternatives and their bioavailabilities, that is the rate and extent after administration in the same molar dose, lie within the acceptable predefined limits. These limits are set to ensure the comparable in vivo performance, that is the similarity in terms of the safety and efficacy. So these pharmacokinetics are related to the pharmacodynamics, and the rate and extent of absorption and the plasma concentration profile is directly related to the pharmacodynamic response, and that is the therapeutic outcome. Then what are the PK parameters? The main PK parameters are AUC and Cmax, which are measured, and also Tmax is required for some of the formulations. So PK parameters are AUC and Cmax. For a pharmacokinetic comparison, the plasma concentration data are used to assess the key pharmacokinetic parameters like AUC, that is area under the curve, peak concentration, that is Cmax, and time to reach the Cmax is called as Tmax. Then absorption lag time. This absorption lag time is there for some of the modified release formulations and delayed release formulations, and also for some of the chronotherapeutically designed formulations. So the Tmax is the time at which maximum concentration is available in the plasma, and Cmax is the max concentration available in the plasma. Testing should be conducted at several different doses, especially when the drug displays non-linear pharmacokinetics. If the drug shows non-linear pharmacokinetics, that time the biostudies are done on the higher and lower strengths, or bracketing studies are done. In addition to data from the bioequivalence studies, some other data is also required by the regulatory authorities, which include analytical method validation data and in vitro in vivo correlation, that is also known as IV-IVC correlations. Then what is the B criteria? When we can say that the product is meeting the bioequivalence criteria or when it is failing. So separate video is there for this criteria, and it is available in the playlist. So for more understanding, that video is very much important. See, as per the WHO, if the 90% confidence interval for the ratio multi-source, that is generic product, and the comparator, that is reference product or RLD, lies within 80 to 125% of the acceptance range for AUC 0 to T and Cmax. So this area under the curve from 0 to T, so T is the specific time till which the AUC is calculated, and this AUC may be from 0 to T or 0 to tau, that is dosing interval, tau means dosing interval, or AUC 0 to infinity. For high variable finished pharmaceutical products, the applicable acceptance ranges for Cmax can be 69.84 to 143.19%, and this Cmax widening is done when the drug formulation shows high variability, and that high variability should be more than 30%, then that product can be said as highly variable. As per Australia, the TGA, that is Therapeutic Goods Administration, considers preparations to be bioequivalent if 90% confidence intervals, that is 90% CI, of the rate ratios between two preparations of Cmax and AUC lie in the range of 0.80 to 1.25, and that is nothing but 80 to 125%. Tmax should be similar between the products. See, Tmax is also required for some of the formulations to show the therapeutic equivalency. The examples are products which are required for rapid analgesia. There are tighter requirements for the drug with narrow therapeutic index and or saturable metabolism. So there are tighter requirements when the drug has a narrow therapeutic index. That means the change in the Cmax will affect the safety and efficacy profile of the formulation. Then as per Europe and US, we will see, as per Europe, two medicinal products are bioequivalent if they are pharmaceutical equivalent or the pharmaceutical alternatives, and if their bioavailabilities after administration in the same molar dose, that means same strength, are similar to such a degree that their effects with respect to both efficacy and safety will be essentially the same. This is considered by demonstrating if the 90% confidence interval, that is 90% CI, of the ratios of AUC and Cmax between the two preparations lie in the range of 80 to 125%. Then as per the US FDA, the FDA considered two products equivalent or bioequivalent if the 90% CI of the relative mean Cmax, AUC and AUC 0 to T and AUC 0 to infinity of the test, that is generic formulation, to the reference, that is innovator or brand formulation, should be within 80 to 125%. And why this all the regulatory authorities considers 80 to 125%? Because as per the regulatory authorities, the difference in the Cmax and AUC of 20% will not give much uh, change or impact on the therapeutic of the formulation. That's why if there is a 20% change, the formulation can be considered as therapeutically equivalent. Then bioequivalence studies. So what are the types of study? There is a fasting state, fed state, steady state or multi-dose studies, and the food effect studies. So fasting stage involves the administration on fasting state, similarly administration in the fed state, and also there is a steady state or multi-dose studies when there is a requirement of inclusion of the patients in the study, when there is a problem of safety, that time these multi-dose studies are required. Multi-dose studies or steady state studies are also required when the formulation shows the deposition into the body, and steady state is required to be achieved to measure the pharmacokinetics. Then food effect studies are also done to study the effect of food on the pharmacokinetics and pharmacodynamics of the formulation. The main pharmacokinetic variables to be measured in the bioequivalence studies are AUC and Cmax. AUC is the extent of absorption and Cmax is the rate of absorption. The 90% confidence intervals is 80 to 125%. Study designs include single dose, two treatment, two period in vivo. That may be crossover design or parallel design. Sometimes replicate studies are also done, and that time the designs are different. The subjects included in the bioequivalence studies may be normal healthy male or female, general population, or specifically male or female, or patients based on the molecule or the drug formulation under the bioequivalence study. Then which are the factors which affect the bioequivalence? Like chemistry of the drugs and BCS class, physicochemical properties, pH dependent solubilities of the API and the formulation, polymorphism and PSD, complex delivery mechanisms which can cause the bioequivalence variations or variances, formulation composition and manufacturing methods, dissolution and IVIVC, and variability in the ADME of drugs. All these factors are affect on the bioequivalence outcome. If the formulation or drug substance shows very much impact on the pH dependent dissolution or pH dependent solubility, then definitely the solubility will affect the bioequivalence outcome. Many factors affect the bioequivalence, like drug related, formulation related, or the patient related factors, disease state factors, then subject related factors. Some subjects are fast metabolizers for the drugs and some are slow. So all these factors collectively affect the outcome of the bioequivalence studies. Then what are the reasons or when we can say that the formulation is not bioequivalent, or it is bioinequivalent, or the bioequivalence study is failed? When there is a lower or higher Cmax, the criteria is 80 to 125, and if this Cmax is lower than 80 and if it is higher than 125, then it can be said that the B is failed. So why this Cmax changes? Because lower or higher in vivo solubility or dissolution rate of the formulation compared to the reference product, then not meeting the AUC. When there is a problem of not meeting the AUC, that means the total extent is less. That time it is said that there might be incompleted dissolution and this dissolution is not in vitro, it might be in the body, that is incomplete in vivo dissolution. The absorption may be incomplete. Here the absorption may be incomplete, that's why the total drug is not absorbed, and that unabsorbed drug get eliminated through the GI, and that's why result into the lower AUC. Degradation in GIT, this is also one of the reason. If the test formulation shows or reference formulation shows more or less degradation in the GIT, then these might not be giving the bioequivalence. Variability in the data due to the subjects, less number of subjects, or presence of the outliers. So this is the also a very important reason for failure of the BE study. The probability of correctly accepting the bioequivalence is the power of the test. If the bioequivalence test is not showing a sufficient power, then the failure or passing criteria cannot be assessed, and all these factors lead to the bio inequivalent outcomes. Then what are highly variable drugs, and what are the solutions for highly variable drugs? So the increase in the subjects for highly variable drugs is one of the main solution, but increasing the subject and getting the bioequivalent data is not always possible. It varies from case by case basis. If there is a variation in the intra subjects for AUC and Cmax greater than 30%, then that drug can be called as highly variable. And also remember here, the more than 30% intra-subject variability should also be there for the reference product. Otherwise, that will not be considered as highly variable drug. Drugs are considered highly variable, but if that drug have a wide therapeutic window, then less stringent bioequivalent criteria can be applied by using the reference scaling method. Also by using the replicate design with 3-period or 4-period crossover. So in this method, the reference scaling is done, and more times the reference product is given, so that the variation in the reference can be made known, and then the test formulation can be statistically tested. But here, when applying this reference scaling and replicate designs, the drug may be highly variable, but it should not have a very narrow therapeutic window. The acceptance criteria for Cmax can be widened to 69.84 to 143.19 from 80 to 125, and this is taken from the Europe BE guidance. Then another solution is adaptive designs and stage-wise BE studies. So adaptive designs and stage-wise BE studies are required to be written into the protocol, and then these studies can be executed. Then also, whenever going to the bioequivalence studies, there should be a firm decision and proper and judicious decision for sample size, and this sample size should be based on the prediction of the variability into the BE studies. So that is anticipated variability. Also, here the sample set should be considered considering the dropouts. So what are those problems for lower and higher Cmax and not meeting the AUC? Mainly the lower Cmax or higher Cmax not meeting the AUC are the probable and the most important reasons for BE failure. So why these lower and higher Cmax values comes, and why the AUC cannot be met? So the main reasons are formulation design and biorelevant or discriminatory dissolution method development, Similarity and differences in the test formulation versus reference formulation. Differences in the in vivo solubility, permeability and stability in the test formulation versus reference formulation. Then BCS class boundaries and solubility regions. Under discriminatory dissolution method and dissolution time point specification in relation to the absorption time. Excipient's role in vivo solubility, dissolution and absorption. Then permeation enhancers, inhibitors and multifunctional excipients. These are some of the reasons for BE failure. So every time whenever conducting a BE study, the formulation design and the differences and the similarities should be written down and its risk should be assessed. Then there should be a availability of discriminatory dissolution method which can discriminate the reference formulation and the test formulation. The test formulation is required to be made similar or inline to the reference formulation so that there will be more chances of meeting the Cmax and AUC criteria, that is meeting the bioequivalence. Then if there is a difference in the in vivo solubility or permeability of the API used in the test formulation and in the reference formulation, then obviously it will impact the Cmax and AUC. For example, if the innovators uses or reference product uses the crystalline API and the generic product uses the amorphous API, so there are chances that the amorphous API will be more soluble compared to crystalline API and the Cmax will be at the higher side. And similarly vice versa can occur if innovator uses the amorphous API and the reference product uses crystalline API. Then the particle size of the API used by the innovator and the particle size used by the generic formulation. Then the excipients. Then changes in the solubility and permeability in vivo and stability of the test formulation in the GI tract. If the test formulation uses the API which is less stable or more stable than the innovator drug, then it will certainly have a great impact on the absorption and will be reflected into the Cmax and AUC. Then BCS class borders and the solubility regions. So generally BCS gives the idea of solubility and permeability, but there are certain limitations that some drugs belong to BCS class 1 and may show the dissolution dependency based on the particle size. So whenever deciding the dissolution criteria, it should be related to the Tmax. Sometimes the BCS class 1 drug or 3 drug may also get degraded into the GIT, and due to which the differences in the degradation in the innovator formulation and the test formulation will have an impact on the Cmax and AUC. Also, the dissolution or discriminatory dissolution should be available, and if the dissolution method used to assess the similarity in the dissolution between reference product and the test product is under discriminatory, then the dissolution time point specification will not be proper in relation to the absorption time, and which will lead to the bioequivalence. Then some excipients are there which have multifunctional role, which also act as disintegrants. Some of the excipients also act as stabilizers. Some excipients improve the solubility in vivo, like povidone is there, which is used as a binder. It also improves the solubility of some of the APIs. Then it also helps to keep the API in the solubilized form. So like these excipients should be evaluated critically. Sometimes the solubilizers are used into the formulations like 280 or SLS, and many others. Then sometimes the permeation enhancers are used and inhibitors are also used. Some multifunctional excipients are there. So all these parameters should be very critically evaluated so that there will be a more chances to meet the Cmax and EUC, which are the most important parameters for bioequivalence. So I think this video might have given you the good understanding of the bioequivalence problems and solutions. So please keep watching the videos and subscribe the Pharma Learning in Depth channel. Thank you.